Good morning, friends. Today is the topic medical microbiology, Salmonella typhi. So Salmonella typhi is caused by the ingestions of food or water contaminated with the Salmonella typhi. So the disease is characterized by fever, headache, abdominal pain, anaphylaxis, malaise, uncoated tongue, and hepatomelalgia, rose spotted on the skin. So typhoid bacilli were first observed by April 1880. Microbiology. So the Salmonella is a gram-negative rod-shaped organism. It's a enterobacterium and measured about 1 to 3 micromillimeter diameter into 0.5 micromillimeter in size. So it is actively motile by peritrachous flagella structure formation. So it is a non-capsulated, non-sporing bacteria, but it may possess the fimbriae. Next one, cultural characteristics. So Salmonella typhi grows rapidly on a simple medium. So it grows over a range of and pH level 6.8 and temperature 15 to 41 degrees Celsius and optimum 37 degrees Celsius. So the colonies are large, low convex, circular, smooth and measured about 2 to 3 micromillimeter in diameter. The colonies are more translucent than coliform colonies. It is anaerobic and facultative anaerobic organism. On mechanic agar medium and deoxycholate citrate medium, the colorless colonies are produced due to the non-lactose fermenting colonies. So the Wilson and Blair bismuth sulfate medium, jet black colonies with a metallic shines are formed due to the productions of H2S production. So it differentiated Salmonella paratyphi A, which do not produce hydrogen sulfide and form a green color colony formation. So next one, biochemical reaction. So Salmonella typhi, the ferment glucose, mannitol, maltose is forming a acid but no gas formation. Sorry. So Salmonella fermenting a glucose, mannitol, maltose and forming a acid and a gas formation. So it does not ferment lactose or a sucrose and do not produce a indole. It means indole negative reactions. So MR methyl red positive reaction, phosphorus negative reaction and citrate positive reaction and urease negative reaction. So next one, resistant. So the typhoid bacilli are killed by the heating at 60 degrees Celsius for 15 minutes to 20 minutes and also by a boiling, chlorination and a pasteurization technique methods. So most of the strains are sensitive to chloramphenicol, ampicillin, tetracycline and extra. So they can also survive in polluted water, soil, ice and snow for a week and month. So next one, antigenic structure. So the antigens are Salmonella are classified into 3 types. The first one, and flagella antigen, H antigen, and somatic antigen or a cell wall antigen, O antigen, and surface antigen, V antigen and Virulent antigen. And first one, H antigen, the flagella or OH antigen is present on the flagella. It's heat-labeled protein and help in acculination formation. So when mixed with the antisera, they acculinate rapidly and form a loosey, fluffy and clumps of acculination. That means antigen-antibody complexes. So next one, O antigen. Somatic or O antigen is a phospholipid protein polysaccharide complex. It is an integral part of cell wall. So it was first isolated by the bovine scientist. So hence it is also called as a bovine antigen. When mixed with the antisera O, antigen form a compact, chalky and granular clumps formation. And next one, VA antigen. Some strains of Salmonella typhi possess the surface K antigen and extended to the cell wall. They refer to as a VA antigen. So it is a heat label and they are not involved in the diagnosis but helpful in epidemiological typing of Salmonella typhi by employing VI bacteriophages. So next, pathogenesis. So Salmonella can follow one of the infection cycle as they are depicted in figure. The one cycle leads to entities whereas the other leads to enteric fever, it means typhoid. First one, the most Salmonella strains causes the enteric. The Salmonella invade the intestinal well and produce the enterotoxin, but there is no spread into the blood. So the two Salmonella serotyphi, typhi as a paratyphi, can cause the typhoid. The organism invade the wall of the terminal ileum and then spread to the intestinal lymphatics when they are phagocytized by the PMNs and the macrophages. Those phagocytized by the macrophages survives and are disseminated via those cells to other lymphatic tissues. So evenly the organism escape the macrophages and are liberated into the blood when they are disseminated throughout the body. So other Salmonella remains within the intestines where they invade the gut wall and many causes of the ulceration, the perforation and the hemorrhages. So Salmonella also multiple within the gallbladder and pear patches. So the pathogenesis of enteric fever, the bacillus on reaching the gut attack the microvilli of a small intestines and penetrate the lamina porifera and submucous membrane. So they are phagocytized by the polymorphics and the macrophages. The bacillus resists the intracellular killing, enter the mesenteric lymph nodules. They enter into the blood through the thoracic duct and causes the transient bacteria disease. So the bacillus are seeded in liver, spleen, bone marrow, gallbladder, lymph nodules and lungs, kidney where further multiplication take places. The bacillus from those are liberated into the blood causing massive bacteremia which helps onset the clinical disease, and finally the enteric fever typhoid caused. So next one, laboratory diagnosis. So the isolations of bacillus from a patient, demonstrations of antibody in serum and specimens. The first week blood culture from the end of 1 week to 2 week viral test analysis from a early stage of disease up to convalescence. The fecal culture becoming a positive reaction, and second and third week of illness the urine culture became positive reaction, and others are the specimens of clot culture, bone marrow biopsy, bile from a duodenal aspiration. So prophylaxis. So the enteric fever, the controlled by the improvements in sanitation and protected water supply, and TA vaccine Salmonella typhi, Salmonella paratyphi, heat killed at 50 degrees Celsius and 50 degrees Celsius to 60 degrees Celsius. At preserved in 0.5% phenol, they given in 2 doses of 0.5% subcutaneously and intervals of 4 to 6 weeks. The typhoid live vaccines, stable mutants of ST strains typhoid 2 lay in entry-coated capsule containing 10⁴ viable liabilities mutants bacillus taken orally 1 hour before food with the water or milk on a days 1, 2, 3, 2 and 5 days. So there are no antibiotic during the period. The typhi VI injectable vaccine with a purified VI polysaccharides antigens of ST strains, TY2 given as a single SC or a intramuscular injection. So treatment, ampicillin, amoxicillin, fluoresceins and carbadoxyls have been found useful in treatments of typhoid fever. Thank you.